All right. So, as you guys know, this is the review one session. So, basically, what we're gonna do is just quickly, skim through what we've learned so far, and sort of, that helps for you to prepare the actual exam, which is this Friday. Before we get to the review, actually, there was one request. I think it's a pretty good one, actually, about the talks for the group called the Doctors Without Borders Student Chapter. So here's an information about her talk here. So you can actually get it in Blackboard too. If you have any questions, contact Abdel, Mr. Abdel Asmarli, who's sitting right there. So you can just, so he's right there. So, you can just talk to him about that. All right. All right. So first of all, we basically covered all the sort of basics or the definitions of the biochemistry, which is part of the introduction. And then we moved on right away to the thermodynamics. So we talked about the first law, which is basically stating that the energy is conserved, so it cannot be created or destroyed. I'm just quickly skimming through. So and then we basically, discuss the definitions of all the energy state functions, such as enthalpy here. And here's the equation for the enthalpy, as you know, delta U, which is the internal energy, plus the delta PV, the work done to the system, right? And this can be converted to basically the QP at the constant pressure, which is the heat value minus worked, and here's also the work has been done to the system. So hopefully you remember all these things or have studied it recently. And then there's a bit of conversion getting us to the delta H equals QP, right? And also at the certain conditions, especially inside the cell, which is a sort of a biochemical environment, the pressure, I'm sorry about that, the volume also doesn't change. So basically it's gonna be delta H, the enthalpy changes is gonna be the change of internal energy, delta U. And we also, covered quite a bit about the Gibbs free energy here and its relationship to the other energy state functions like delta H, the enthalpy changes, as well as entropy changes, the delta H. And also there's a relationship here. And depending on the what value you have for delta H or delta S, you have different results for Gibbs free energy. And that also leads us to predict to that whether this reaction in interest is spontaneous or not, right? And we use the different terms for that, like endergonic, exergonic reactions. And also we talked about other terms like exothermic, endothermic processes and things like that. So you should be able to distinguish those different terms, for the stating different reactions, and whether some of these are related to the spontaneity of the reaction or not. So here's a relationship again that was basically driven by this relationship, which you can get in the sort of as the related to the temperature here in Kelvin. And here's a quick summary for the thermodynamics, basically. So everything about the spontaneity of the reaction, and you can tell that by the delta G value, whether this is negative or the positive, and have different terms to call them. And the delta G0 means that it doesn't mean that the reaction is just on hold and doesn't do anything, but it actually means that the reaction reaches at the equilibrium. So everything's pretty dynamic and active still, but you don't see the overall changes in the concentration of the reactants or the products in this case. And it's all about the spontaneity itself, the direction of the reaction, but not about the rate of the reaction. And we moved on a little bit from that and also used the KEQ constant. Basically, this is the dissociation constant, as you know, at the equilibrium. And we discussed the relationship between the delta G and KEQ. Here's a definition for KEQ. And I gave you an example basically using the Gibbs free energy here, how these values change and relevant to KEQ. There's a relationship here, which is really important, right? And delta G naught is basically delta G, the Gibbs free energy at the standard state. And we also discussed what the standard states are and how they're different from chemists' standard state and the biochemists' standard state. And keep going on this relationship to get basically derive this equation here, so we can directly relate the KEQ to delta G naught here. Question? That is a good question. So in theory, I mean, you can put any kinds of concentrations there, right? But we're talking about delta G now, which is at the standard state. And everything, I mean, is pretty much like a changing, but you can't really, I mean, discuss anything with the changing values. So what you typically do is that you just let the reaction go, and then just wait for a while to get the equilibrium. And then now you can get the actual value out, right? Otherwise, delta G is changing over time, every time. So you can't just, just define it as a one value, as opposed to when you look at delta G or delta G naught values everywhere, that just is under assumption of the reaction is at the equilibrium all the time. That is a really good question. And that really is true, right? So, I mean, how can you solve this problem? I think it's a really good point, actually. So, any speculations? Any of you guys? Since let's just assume that we studied a little bit on this, And we talked about the KEQ, basically the concentrations, the ratio of the concentrations of the reactants and the products at the equilibrium. And now we're talking about the delta G values, right? Which is supposedly at the equilibrium. And isn't it supposed to be zero then? This is equilibrium, right? What do you think? Any? So it depends on the temperature, right? So whenever you change the temperature, this value changes. So this is definitely a constant value at the certain reaction, and that'll reach to zero basically at the equilibrium. But we have different different reaction that is added to this. So it's always the state of the combination of the temperature too. So there's another variable, temperature is involved, but that's why we come up with this value, basically to drive it. At the any kinds of temperature, this G values, of course, G values changes, delta G changes. So this is looking at the relationship between the KEQ and delta G, but that also relies on the temperature. Does that make sense to everybody? So that's why here too, we have KEQ and delta G naught. So this should be just a constant value, okay, zero or whatever that is. But there's another state function, which is temperature. So it has to be basically normalized by the temperature. All right? Okay, so this relationship is really important because basically we can use that relationship to get the Banach plot right here. And Banach plot is basically, getting the delta H or delta H naught or the delta S naught by having the relationship between the KEQ and temperature. Again, temperature changes, right? So that's why KEQ value changes too, and that affects the changes in delta G. All right. So the slope, with the slope, you can get the basically delta H naught value, as opposed to with the intercept, you can get the delta S naught value, which are hard to measure, with the different methods. All right. So here's a quick summary again about the thermodynamics. Delta U, which is heat minus work. Delta H, we talked about this. And the entropy, the definition of the entropy is here with the equation. And from there, we derive the Gibbs free energy. And basically talked about what Gibbs free energy means, positive, negative values, and so on, as well as relationship of the KEQ and the delta G naught here. And this delta G of the reaction is basically additive, right? So you can basically look up all the tables where it lists all the delta G values for the all different kinds of reactions, and you can just pick those values, find those values, and then sum up to get the total delta G value. Any questions so far? Delta G naught? Yes. So the delta G naught is, no, I'm sorry about that. So delta G naught is basically delta G value at the standard state. So that's where the reaction is just a set. And then there's gonna be a constant rate of reaction going on. And that talks about the energy to drive that. It's not about the how much of actual energy that is input or output. It's only the measure, sort of arbitrary number that we can either tell that this one happens naturally, spontaneously or not. That's all, right? So even if it's at the equilibrium or not, you're gonna have delta G value. And it's delta G naught is basically at the standard state. That's all. Exactly. So delta G is going to be 0 at the equilibrium, right? But you have a set delta G naught value, which is not 0 there. And you have RT basically, ln KEQ value there. No, actually right here. RT ln, KEQ right here. Which, depending on that reaction is, you can have a positive, all the positive values, but you can get the positive value or negative values because it's log, basically. All right. And then we moved on from there, thermodynamics, and talked about different kinds of bondings, like a covalent bonding, non-covalent bondings, and basically spent quite a bit of time to talk about the interactions or the energies that is involved in water molecule interactions, right? So here's a covalent bonding, which ranges like here, 300, 400 or so. And there are different types of non-covalent bondings, including ionic interactions, hydrogen bonding, dipole-dipole, London dispersion, which are all under the category of van der Waals forces. And then again, of course, we talked about hydrogen bonding, which are the donors, which are the acceptors, and the requirements for being a donor, acceptor, and things like that, which are really important. And talked about the dipole moment as well as electronegativity of the atoms, specific atoms that can work as either donor or acceptor, or the helping the donor, which is a hydrogen atom. Okay. And we also talked about the definition of solution and more definitions, about the solvents, colloids, suspensions, and their differences and things like that. And talked about how we calculate the percentages or the molar concentrations or so. And there are all different ways, like a volume to volume or weight to weight and so forth. And also because we talked about the dipoles or the water molecules, which is polar, we further discussed the polar substances or polar molecules and non-polar molecules and things like that. They have, of course, depending on their charges or the dipole moments they have, they can be categorized as the hydrophiles or hydrophobes or amphiphiles. And they have like particular properties, right? basically, this is not about, just me like explaining again about these concepts, but more like the quickly covering all the topics that you need to review. And because we discussed the solutions, we also talked about the colligative properties, which is have these interesting features when you have this some solute with in the solvent. So basically, whenever you have solutions, you're gonna have these colligative properties, and this is not dependent on the types of molecules or the particles, but it depends only on the concentration, the number of molecules in the solution. Talked about osmotic pressure, and also how the animal cells or the plant cells overcome this osmotic stress. Moving on, and we talked about the dialysis too, right? Which I didn't include here, but you should know what it is, what it means, and how it is used too. And then since we talked about the water, we also talked about the equilibrium constant, or the dissociation constant at the equilibrium for the water molecule, how it is ionized, which is pretty rare, pretty little amount, right? And you could basically get Kw, which is ionic product. And then further again, we use this ionic product to get the pH, which is really useful to the measure the acidity of the solution, right? Basically the content. And there was a reciprocal relationship or anti-correlation basically between the hydrogen cation concentration and hydroxide concentration. We also talked about the acid-base reactions as well as their definitions using Bronsted-Lowry view here about either donating or accepting protons, right? So even the same water molecule can be work as base or acid based on their partner for any reactions there. And it's all about the protons, basically, the positive charge, where it goes and how it is accepted. And we also talked about the acid strength, which is estimated by the Ka value here, or pKa, and Ka is basically a dissociation constant for the acid, right? And pKa is just getting the minus log of that value with the base of 10. So it's quite looking similar to pH, right? And of course, we can make a relationship between the pKa and pH value under the condition that this conjugate base and the acid concentration is known. And it's called the Henderson Hasselbalch equation, where we actually went through each steps here to get this relationship. And under a certain condition where this value becomes 0, so that means the conjugate base, concentration of the conjugate base is the same as the concentration of acid, the log function, the logarithm will be 0. So you're going to basically get pH equals pKa. And we talked about the titration curve too. Basically, use the either weak acid or weak base, and use the strong base or strong acid to neutralize those acid or base, right? And that's the process. And the requirement is that always you use weak acid or weak base, starting with them. And when you titrate them, as you can see, you see this kind of nice sigmoidal kind of shape, and the ranges of change is pretty much the same between all different kinds of acids or base, but only the changes, the differences will be the where they are located on this pH scale. So you can just move up and down like that depending on which acid you use or base you use. And interestingly, right in the middle, right here, the inflection point would be where pH equals pKa. And as we talked in the previous slide, it's basically where the conjugate base concentration is the same as the concentration of acid. Okay? I know it's kind of monotone and kind of going really quick, but still keep in mind that these are important concepts, not only for the exam, but for the anything relevant to biology. And we talked about some buffering system, and an example that we took was basically blood buffering system using the carbonic acid and bicarbonate. Here, the reactions here, basically, those are two different reactions that are connected to each other, right? And then whenever there's one thing is skewed, like going down or going up like that, you basically compensate it by putting the other one a lot more, right? Getting to the equilibrium. And these are the possible disease that can occur if one of these reactions doesn't occur properly. Yes? No, I'm sorry. I thought you were pointing out something and then so. All right. And then we talked about the really critical biomacromolecules, especially nucleic acids. Those are DNAs and RNAs. And we talked about their functions and structures and their subunits, which are nucleotides, and the actual structure of the nucleotides too, that have three different parts or the components, right? Which are the nitrogenous base, sugar base, basically the pentose, and the phosphate group here, which can be mono, di or tri monophosphate phosphate. And talked about the backbone, the property of backbone that is negatively charged because of the phosphate group. And you should be able to distinguish between the nucleoside and nucleotide. It's just only the difference of inclusion of this phosphate group or not. And the nomenclature basically for that based on the IUPAC too. And again, you should be able to at least distinguish and identify those different nucleotides based on their shape, structure. Okay, and these are some shorthands. Again, these are the structures based on what you have attached to the this and glycosidic bond. Oh, sorry about that. On the this phosphate group, you can have different names like that, monophosphate and so on. And also, we talked about the actual interactions, the forces that are involved in these base pairing. So between the C and G here, there are 3 hydrogen bondings that are involved there, and these are the locations. And we also discussed that which are the acceptors and donors and things like that. Same thing applies to the T and A base pairing too. And we talked about the molecular cloning, why we do that, and what kind of things you can get, and what kind of proteins or enzymes that are involved in these processes, and how you can generate the sticky ends or blunt ends and things like that. How you can fuse them together, basically using ligases and so on. And also hyperchromic shift or the effect, which is basically the UV observance of the DNA molecule that changes depending on their structure, whether the DNA is denatured or having that double strand structure. And also we talked about different types of techniques to detect the different macromolecules like the DNA, RNA, and proteins. So southern blot, northern blot, and western blot. And then we delve into more details of the DNA structure now as a polymer here, and talked about this double helical structure as well as major minor groups and their features and so on. And also where the backbone is located and the center of that, how well structured they are without any kinks or bulges or anything like that. And we also talked about the different forms of DNA helices, under which conditions these forms are preferably exist. B form is basically sort of a normal form that you can see. It's a Watson-Crick model, basic model. You can see it most of the oftentimes, in any kinds of physiological conditions where you can see the clear major groups and minor groups. Right here, I'm sorry. As opposed to A form as a sort of squashed and down form, right? So the distinguish between major and minor groups are a little like subtle now. And also they have like a larger diameter, shorter distance and stuff like that. And basically C form is the other way around. And it is also left-handed and can be used in different cases. Here was the quick summary, what kind of bonding or the interactions that are involved for stabilizing the double helical structure of DNA, or even DNA, RNA, or double-stranded RNA too. And I promise you that I'm not going to ask anything about the direction of the strand or anything, but you should know that everything is 5' to 3'. Pretty much like, elongation and everything happens like that way. Elongation, I'm talking about the DNA replication. And also when you look at the double-stranded DNA, then it should be anti-parallel. So one goes, from top to top, 5' to 3', and the other goes to 3' to 5', and so on. And also the hyperchromic shift is again really important, and this cooperativity that you can see on during the hyperchromic shift can be applied to it both formation and destruction of DNA double helix. And talked about the stacking energies that can be different based on the what kind of base pair you have. And we talked about prokaryotic topoisomerases, different types of topoisomerases, and what are the features of them right here, right? And how they work too. And we moved on from there and then talked further about the sort of higher order of the DNA structure, which includes the nucleosomes, and the bigger structure would be 30 nanometer fiber or chromatin fiber, and the final packaging as 300 nanometer fiber or 700 nanometer fiber, depending on which cell cycle stages that you're looking at the chromosomes, and basically the chromosome at the most condensed form. And we talked about packing ratio too. And also talked about the RNAs, the types of RNAs, their structures, and the importance of having secondary structures like helix, loops, bulges, and so on there. And they can make them sort of enzymatic functions and so on. So that's pretty much it. And what I'm planning to do right now is to go over these practice questions. And I'm not going to dwell on this, right, basically, but quickly just to go through that. So this is already uploaded in Blackboard, so you definitely can just, look at it if you feel confident that you got all the answers correct, then you can just leave. That's completely fine to me. So, in case of someone's, confused with anything, I want to just to go over and make sure that everybody's on the same page. So the first one is pretty simple, right? I could just ask, the right answer to any of these people, but for this one, for the liquid water ice structure, what is involved there? I mean, it's just so clear that it's hydrogen bonding, right? And the second one, at pH 10, what would be the concentration of hydroxide? Can anyone explain this? Yes. What is it? Exactly. So then you do 1 times 10 to the negative 10 times concentration of OH equals 1 times 10 to the negative 14. So we get 1 times 10 to the negative. Exactly. So I don't know if any of you actually heard this, but if you guys have any questions, just don't be shy. You can just raise hand and ask questions here. But basically, what I wanted to ask here was the relationship between the hydrogen cation concentration and the hydroxide concentration. And you should be able to use that in the relationship of ionic, what is it, the constant, right? or KW. So you should be able to do that. Here, this can be a little confusing or a little tricky to answer. A reaction that has delta H positive and delta S positive. And of course, we're talking about whether this is endergonic or exergonic, right? So it's spontaneity. So you have to, getting at that with the delta G value, whether this is positive or negative value. To do so, you can either just simply memorize the relationship of that, which is delta G equals delta H minus T delta S, right? Or you can just understand the different relationships and what it means in terms of delta G value, or you can just simply derive it from that equation that I talked to you earlier, right? So again, delta G equals delta H minus T delta S. So you can just rearrange that relationship a little bit to get the T equals delta H divided by delta S, right? And basically, depending on what direction that you're looking at, it can be either exergonic or endergonic. Make sense? Just stop me whenever you have any questions. Weak acid is basically some substance that gives out a proton, tend not to give proton too much, right? It gives out a little bit, but not too much. So it's gonna be what is partially ionized in aqueous solution. Make sense? So it releases hydrogen cation or hydronium, basically, but not much. Heat denatured DNA exhibits increases UV absorbance, and that is cold. Well, there are different things, but basically we talked about hyperchromic effect here, right? It's all about UV absorbance and how it is changed depending on the structure of double helical DNA structure, DNA, right? So, and of course, melting temperature determines that structure of DNA. So heat denatured, meaning that they're loose now, or the single-stranded DNA. So they're gonna have the higher absorbance here, which is directly stating the hyperchromic effect. Polymerase chain reaction, PCR. I hope all of you know this, but is basically just amplifying the DNA fragments. And this can be a little, again, tricky question here, but which will be the conjugate acid of H2PO4 minus, basically, the dihydrogen phosphate. And you can just think of it, since this is a conjugate acid, it has to be derived from base, right? Which is matched directly with the conjugate acid. And base is basically one that accepts the proton, right? So have to have another proton there. So it's gonna be B, which has one more proton than this conjugate acid. Does that make sense? All right. And blah, blah, blah. This is pretty long, but the nucleotide and the basically how they're connected to each other. The nucleotides are connected to each other to form the polymer, which is DNA, and what kind of bonding is involved there. So you can just read it here, but these are pretty much like sort of like make you statements that try to make you confused. The D is the right answer, as there's only one phosphate group is involved, right? And this phosphate group is esterified, meaning that it has a single bond through oxygen atom to the both nucleotides. Make sense? What's the major group, the property of a major group of a bDNA? Again, you should know what bDNA is, which is a sort of a normal like DNA structure under a physiological condition, and that the major group of that should have the wide and deep major group, and the sort of narrow and shallow minor group. Here we're talking about the molecule movement from the one chamber to the other, right? Sort of diffuse out. And what is involved there is pretty much like entropy, which is a measure of disorder of the molecules. Any questions so far? Here, I mean, as long as you know the relationship between the delta H and delta S to tell if the reaction is endergonic or exergonic, which means that spontaneous or non-spontaneous, you should be able to answer this question. So the right answer is going to be negative delta H and negative delta S. And you should go back to that table that shows the relationship of these and the different combinations and so on. Spontaneous process, of course, it's all about delta G. Everything, right? Exergonic. I could have just written as exothermic, right? In which case, that would be incorrect. Van Hoff plot basically, shows the relationship between KEQ and delta G naught, right? So it's not T, it's not minus T delta S or anything, should be able to, tell the relationship and what it means too. It actually shows the relationship of the delta KEQ and the T inverse T basically. Sorry about that here. KEQ and versus inverse T to estimate the delta H naught and delta S naught. Yes. That is a really good question because it's not ln. So please be careful with these kinds of things, That's why I actually made all my questions in 12 font size 12. So hopefully you don't miss anything like small letters and things like that, because we're gonna have a lot of superscripts and subscripts and things like that. So careful. Here's a question about the KEQ and under the standard state and the relationship with the delta G naught basically. So you can either derive it, derive the equation from the first ones, or you can just memorize it as it is right here. And the strongest non-covalent interactions are, what's the answer? Ionic interactions, right? It's much stronger than other interactions. And the second strongest interaction would be which one? Hydrogen bonds, that's correct. And then the weakest one will be London dispersion forces, which is induced dipole to induce the dipole interaction, right? And then the Waals forces are including basically B, C, and D. Make sense? Is ionic interaction under van der Waals force? No. Because van der Waals force is all about the dipoles, right? Permanent or induced dipoles, as opposed to ionic reactions are for the permanent charges, charged molecules. Which of the following statement about water is not true? So you can just read it about everything. But it can form 2 hydrogen bonds per water molecule. How many? 4, up to 4. Blah, blah, blah. Oh, yeah. 16. Yes. Yes. Yes, it is correct. So it has to be tetrahedral. Oh. But it can, right? So the water molecule itself, the molecule itself make the tetrahedral structure. But the interaction of water molecules can be occur between 3 water molecules, 4, 5, or 6, which is a hexagonal shape. In which case, hexagonal lattice, meaning that there is a flat plane with the 6 water molecules are involved for hydrogen bonding. Make sense? So it's different than the molecule structure itself. So this is fairly, straightforward too, right? So the what is it? Methanol and ethanol are pretty much stable and soluble in water because it can also make the hydrogen bonding because it has the hydroxyl group. Okay, so any ethanols, as you know, you can just mix it and get the pretty much like 30%, 70% of ethanol is like our everyday life now, right? You just have to wash hands with it, and this is basically a pure solution because hydrogen bonding, so you can just fuse it in and perfectly be a solution. Hydrophobic interaction that is not true is going to be A. So it says they are caused by hydrophobic molecules interacting to each other. How come it's wrong? There's no force built up there. Time to time, there could be a sort of dipole moment that is built up there. You can imagine that, right? These are just a nonpolar substance. But at the same time, you can just build up a little dipole moment, so induce the dipole and should be able to interact with each other, right? It is absolutely possible, but rarely happens because the hydrophobic molecules are typically really bulky, really big. We're not talking about just a single small purified chemical compound. We're talking about the big block of certain things, like a big protein and things like that. So that's why typically it doesn't involve any kinds of intermolecular reaction between the hydrophobic molecules. Yes? Why the entropy is driven? Can anyone actually explain why the entropy is driven? Quickly, just to checking other people. Does it make sense? Try to find the marker here. So you have to, of course, there's no tip. 2 hydrophobes here in water. So there are a lot of water molecules, right? Something like that. And it's basically making clathrin cage-like a structure, like that. And what you can count the number of water molecules that are interacting with hydrophobes are these numbers. Let's say that those were 20 or so. So you actually have 20 water molecules that are sort of unstable. They're excited a little bit. They don't like that state, but just they have to be like that. As opposed to this one aggregates. Let's say that these are the same size. And then you're going to have a much fewer because now you're getting rid of the water molecules that are involved for stabilizing this, molecules between this, right? So you're going to end up having a fewer molecule that are exposed to hydrophobes. So entropically, this is more favorable. Make sense? That is absolutely right, actually. That's a great point. So when you look at it, when you focus on these hydrophobic molecules, they are aggregating. So their entropy should go down, right? Which is sort of unnatural. But in terms of water molecules, they are more like spread out because they have to be trapped in here. A lot of them are trapped in here as opposed to much less are trapped in here. And all of them are just pretty much spread out and making the hydrogen bonding. So water molecule wise, and this is the whole system. Make sense? So it's all about not just the one type of molecule interaction, but the whole thing in the system, right? So when you add everything up, then that's gonna be negative, driving the negative. And of course, there are another energies that are involved. The hydrogen bonding itself, it just prefers to have a hydrogen bonding with the other molecule as a donor acceptor, right? And that is, there are more molecules that are involved with that reaction, which stabilizes all this state. So it lowers the internal energy, right? So the internal energy is going down. So that's another factor. Okay. All right. So the next one, you see the two layers of amphiphilic molecules and stuff like that. As you know, this is bilayer membranes. And another important thing is the micelles, right? Which is with the single layer of amphiphiles. And instead of they're making the really nice kind of flat layer, they're making the like blob, sphere, right? And as you know, the exposed, the side that is the water molecule has to be amphiphilic. Osmotic pressure is about, which one is it? Concentration. It's all about concentration. And here's a Kw, ionic product of water is 10 to the minus 14th, as you know. And that has to be at which degree? 25, which is the standard state. And it's the same for the both chemists and biochemists definition for the standard state. And here, this is a similar question that I've asked earlier about that acid-base reaction, basically. But this is about the polyprotic acid, where it releases the proton multiple times during the course of titration, basically. And these are the pK values. And when this substance is at the pH of 9.3, which one it'll be? So 9.3 is sitting right between these two, inflection points, right? Or the pK values. So that's gonna be the form right here. So you should know the phosphoric acid shape. What is it? The formula, and you can just calculate back. But basically, the most basic form, the form that lost all the proton would be just a phosphate, right? So this is just a phosphate, PO4 3 minus, right? Sitting if the pH goes above the 12.38. And then when it's sitting right in between, then it's gonna be the form that have one proton accepted, right? So that's why it's gonna be HPO4 2 minus. So you don't even need to memorize this. As long as you know the most basic form or the most acidic form, then you can always just add one proton at a time, when it goes through jump through that inflection point where pH equals pKa. Make sense? I hope, to make it simple. All right, again, pH in the titration curve of a weak acid, would be where the pH equals pKa, that's gonna be the midpoint, the inflection point. This is particularly important when you're doing actual experiments, right? Whenever you make I don't know, some of you guys who have experience with actual research in the lab probably have made some, media or reactions or things like that. And in the end, of course, of making the media, you always, control the pH. Just measure the pH and add the either like sodium hydroxide or the hydrogen chloride and things like that. And that pH is important because of the solute that's getting into that solvent, because you have to have the right pKa value or the pH value, to get the reaction go to the right direction and also find the right partners for the as a reactants. Nucleotides, nucleotides store genetic information. Isn't it true? nucleotides store genetic information. Why not? I'm looking at you, yes. So, because I saw that you're shaking your head. So, anyone? Why? Oops. Exactly. I mean, there's a nitrogenous base. I mean, that's something that's meaningful, that's different than other nucleotides. But with just one thing, that's not a genetic information, right? That's just a different entity, that's all. You have to have it in a string, as a sequence to make it, as something meaningful, like genetic information. Chargaff's rule, although we didn't talk about it, during this review session, you should know what that is. That's basically A equals T, C equals G, and so on, right? Tautomers. What are the tautomers? Can anyone explain briefly? What are the tautomers? Yes. I'm sorry? Exactly. So that basically have only difference in conformation. So that's why it's called constitutional, right? Isomers. So they are having the exact same components or constituents, but arrangements is different. So got it right? So talking about, asking about the tautomers, especially for the nucleic acid. Who can answer who can answer this question? What's the right answer? A, how come? That is correct, but how come? Perfect. So I don't need to explain further, basically. You can just, look at the nitrogenous base structures and think about, where the hydrogen bonding can make, and then there's always blocking from that hydrogen atom that is attached to the oxygen. So it has to move away to basically serve as an acceptor of the hydrogen bonding. So when you look at the pictures of the base pairing, it'll make more sense. Yeah, basically, I mean, I hate this kind of question, but what can I do? I mean, it's just structure. The right answer is D. And of course, you should be able to identify these different shapes. Which base is not present in RNA? Is it adenine, cytosine, thymine, right? So you're basically looking for the thymine. Somebody might be confused with the uracil. Oh, it has to be uracil, and then just to put it, incorrect answer. So be careful. It is impossible for RNA to store genetic information because what would be the right answer? What is it? E? Oh, yeah. So RNA also can store genetic information because in the end, I mean, that contains a sequence too, right? And think about the RNA world hypothesis where RNA itself can be used as a genetic carrier, right? Genetic information carrier, like an RNA virus. So you can always convert back to DNA or the protein and things like that. Molecular cloning and so on. And you want to select the something that is already transformed, meaning that having the meaningful intended DNA fragment in that organism. And how to select it is basically use an antibiotics or the antibiotic resistance gene or the selection marker and so on. But you should be aware of, I mean, not aware, but you should know what these other things are and how they are used for molecular cloning, like restriction sites for inserting certain foreign genes and so on, or the origin of replication, basically to make this plasmid or the vector that you're generating is recognized by the polymerase in the organisms that you're using, like yeast or bacteria, and should be able to go forward and just to replicate again and again, right? So that's important. The last question, for PCR, what are required? What's the right answer? A. So as long as you have these components, you're going to actually make the PCR going. And you should be able to tell what the NTPs are different from the NTPs and so on. That's it. So whenever you have questions, just email me, or you can come to the office hour. And also, we're going to have the exam on the coming Friday, the same time, 11:40. You're going to be given, 1 hour and 20 minutes, basically. Good luck. Hey, just a second.